So welcome to our annual Texas A&M University Corpus Christi 3-minute thesis competition for master's and doctoral level students. On behalf of our presenters, in the first couple rows, and the College of Graduate Studies, I thank you for coming out and showing your support. Go into competition. So let's take a look. Okay, so contestant number one is Quan Nguyen. Okay, his program is getting a master's in mathematics. His advisor is Pete Zimmer, Dr. Pete Zimmer. Okay. Contestant number two is Rachel Wyson. Rachel is getting a PhD in marine biology with her advisor, Randy Reese. Contestant number three, Jeb Struder, is getting his Master's of Science in Kinesiology. His advisor is Dr. Heather E. Webb. Contestant number four is Kimberly Mincy. She's working on her PhD in counselor education. Her advisor is Michelle Hollenbaugh. Contestant number five is Julia Delacqua. She is getting her PhD in counselor education. Her advisor is Stephen Lenz. Contestant number six is Tomas Espinosa, who is working on his PhD in curriculum and instruction. His advisor is Corinne Valadez. Contestant number seven is Vipin Sharma. He's getting his Master's of Science in Environmental Science. His advisor is Dr. David Felix. Contestant number eight is Jamu Bahula, okay, who is working on his Master's in Accounting. His advisor is Dr. Donald Deese. Contestant number nine is Megan Mullis, who's working on a PhD in marine biology. Her advisor is also Brandy Reese. Contestant number 10. We have a lot of contestants. Is Rochelle Cortino, who's working on her PhD in curriculum and instruction. Her advisor is Dr. Bethany Fletcher. Contestant number 11 is Lauren McGregor, who's receiving her Master's in Science in Computational Chemistry. Her advisor is Dr. Tim Cosgrove. Contestant number 12 is Jan Brott. Jan Brott's working on her EdD in Educational Leadership, and her advisor is Kabiar Kuzakanani. I love saying that name. Contestant number 13 is Hongming Yao, who's working on his PhD in Coastal and Marine Science, System Science. His advisor is Xingping Hu. And last but not least, Christian Cunningham, who is working on his Master's in Marine Biology. His advisor is also Brandy Kyle Reese. She is busy. Okay, so let's give a hand to all the contestants. And we'll get ready to start the competition. His presentation is entitled Inferring Networks for Graph Partitioning.
Hello everyone. So I'll talk about my master thesis. It's about an inferring network for graph partitioning. The main application of my thesis is in image segmentation. You see, if you have an input image, the goal is to segment that image into meaningful regions. This image segmentation is usually applied at first step in many computer vision systems. So, for example, in medical imaging, we can use this as first step in locating tumors, blood veins, organ structure. Like in Facebook or Instagram, we can use this to locate where in the image is the face of a person, and so on. So we, in our thesis, we view an image as a graph where each vertex, you see the each vertex will be represented as a pixel, and there will be a link between two vertices. The link will represent the similarity between two vertices, and we will use a neural network to assign a correlation score that represent how quantify how similar two vertices are. Okay, so if the link is strong, then they should be in the same segment. They should belong to the same object. And once we define the graph, then we can start cutting this graph. By cutting, I mean discarding edge that is below the threshold. Okay. So for example, if by doing this, if a link is too weak below threshold, we separate the two vertices, the two weakly connected vertices. So the result will be the segmentation like you see in the right-hand side. The novel in my thesis is that for each threshold that you choose, there will be a cost to it. So there's different way to cut the graph using the threshold, right? So by varying this threshold, we will have a different energy, different cost, and we will try to minimizing this cost in our training process. By doing this, we are able to achieve 5% increasement in performance compared to using a fixed threshold in our training process of the neural network. So neural network will look at a bunch of example of image and their segmentation, ground truth segmentation, and the neural will be learning from there. That's conclude my talk.
Your title of talk is M3, Mangroves, Methane, and Microbes.
All right, hi everyone. So the premise of my research today is M cubed, mangroves, methane, and microbes. And these are all three very different from each other, but they tie in together very nicely and kind of tie in together in this looping fashion. But one of these is you can actually see. The other two are invisible to the naked eye. The top, we have a picture of the mangroves. The bottom, we are depicting our methane with the image of a flux chamber, reading in real time concentrations of methane fluxing out of the sediment up into the atmosphere. And then we have, over to the side, our microbial population, something that's often forgotten in research, but after hours of lab work and work on the computer, we can begin to understand their genetic information and their structure and function in these systems. And these all three tie in together, but to understand how they do that, we need to understand them piece by piece. So to begin, mangroves, again shown on the top right, are these small wetland plants that line the Texas coastline. They are home to birds, fish, butterflies, and they're often recreation sites for humans. And with fewer winter freezes, these mangroves are actually migrating up the coastline. And while that's great, it's going into other places such as Spartina, a seagrass vegetation sites. But another benefit to mangroves is that with their thick, carbon-rich sediment, they're actually protecting us from big coastal storms. And that carbon-rich sediment is where we get our methane production. This methane gets produced from the carbon source and goes up into the atmosphere. Methane, a greenhouse gas, then traps heat in our atmosphere, leading to the increased loop of global warming. And then we have our microbes, which you may be wondering, well, how does that fit in with this? Well, so those microbes are actually creating that methane as their energy source. So they're taking the methane and producing it, or taking carbon and producing methane. Alternatively, there are other microbe populations that take the methane and have an energy from it. Now, all of these kind of loop back and forth together. So when you talk about one, you kind of lean into the next topic. So with increased winter temperatures, we have mangrove migration. With mangrove migration, we have more carbon-rich sediment. With more carbon-rich sediment, we have more methane production by microbial respiration. And by microbial respiration, with more methane production, you have this impact back into the global warming cycle. But one of the things that I really enjoy about this project is we get to look here. We're looking at this small pit that is tying into the rest of the project. And what if they're playing a bigger role than what we thought they were? What if for every molecule of methane produced, 2 are being consumed? What if these communities are so changing that they can alter between day and night or season to season? And that's why I like this. It makes microbes matter. Thank you.
This presentation is entitled The Neuromuscular and Muscle Damage Responses to the Farmer's Walk.
It's a battle that's listed at the end of time. A worthy adversary for those that choose to accept it. You've gotten your food, you've given your money to HEB, and you've driven back to the privacy of your own home. The only thing that stands in your way is the treacherous adventure of taking your groceries from your car into your house. Now, I don't know about you, but I will only be taking one trip, or I will die trying. Now, what if I was to tell you there's actually a functional exercise that mimics these exact same stressors, but it also provides improvements in balance, strength, and lower body power? Well, that's what known as a farmer's walk, and it does just that. Now, the farmer's walk is actually an example of a strongman training exercise that's been introduced to numerous fitness environments, from our Islanders over at D1 Athletics to our local fitness and rehab facilities here in Corpus Christi. Now, previous investigators looked at differences in walking mechanics, patterns of muscle activation, and differences in performance between actual athletes and Joe Schmoes like me and you. However, what I've found is there's a lack of biochemical and cue response information to the farmer's walk. Are we injuring ourselves? Are we sore the next day? Is this torture really worth it? Well, that's where I come in. You see, I wanted to measure if there were sustained muscle damage biomarkers via blood draws, performance decreases via vertical jump height, and perceived muscle soreness via survey. So, I gathered a group of lovely Islanders, brought them back to my lab, and we measured their baseline measurements of vertical jump heights and their maximum weight they could lift via deadlift. We then took 7% of their weights, had them walk for 20 meters for 10 total repetitions. And after this wonderful experience, we brought them back to my lab, and we took their blood draws at 6 different time points and their vertical jump height at 3 different time points over a 3-day period post-exercise. So, what did we find? Well, apparently we had nothing to worry about. There were no differences in muscle damage responses, and there were no differences in performance decreases when compared to the control group. However, we did see differences in perceived muscle soreness. So what does this mean? Well, this is the first documented study of an acute biochemical response to the farmer's walk at this intensity and this duration. We did see differences in perceived muscle soreness between the upper body and the lower body. We believe this may be from actually carrying that weight in your hands for over 200 meters. But at the end of the day, the message I want to get across is this is a functional exercise for everybody. From our students walking across campus with their book bags, faculty members with their supplies, even firefighters and military personnel can benefit from this exercise. And to top it all off, we had a partner study that actually saw we had increases in energy and decreases in intensity, or decreases in tiredness after performing the farmer's walk. So, you don't have to be a strong man to bring the groceries. Core exercise physiology will show you why. Thank you.
The title of her talk is A Qualitative Inquiry on the Lived Experiences of People Who Engage with Pro-Eating Disorder Online Media.
When you think about eating disorders, you might imagine a group of high school girls sitting in the corner drinking diet soda in a cafeteria. You might also envision someone who decides to leave after they've eaten and go to the washroom. You also might consider a young woman who says she's just not hungry after she's eaten. These are pictures of what society has told us eating disorders look like. It's really hard for us to consider that pain and that loneliness that people experience when they have eating disorders. It's hard for us to connect to that misunderstanding that they often feel. In my study, I aim to look at the lived experiences of people who use a form of social media called pro-eating disorder online media. These web pages allow people to freely access and share information related to eating disorders and develop deep and meaningful relationships. To give you further context, I'm going to give you a story about one of my participants. In my study, I used semi-structured interviews, and I reviewed the post-history content of 10 community members across 5 different countries. Abby's 31, she's from the United States, and she's had an eating disorder for over a decade. When Abby first found pro-eating disorder online media, she wasn't that interested in it. But she kept Googling and she kept searching, and eventually one day she was hooked. My participant said that experience was like falling down a rabbit hole. After she was immersed, she felt accepted and understood. She belonged. She had a community. That's not something she experienced before. She learned a lot about herself. She learned a lot about her eating disorder, and she also learned ways to reduce harm when she was performing eating disorder behaviors. However, there's a lot of consequences. Abby had an eating disorder, and it told her that she didn't deserve recovery. She believed it. She hated the idea of leaving her eating disorder and leaving these communities. They were her home. There were a lot of triggers, though. There were a lot of consequences. So Abby had a really fortunate experience where one of her community members encouraged her to seek recovery. And so she did. Sometimes it's a good thing and she's really hopeful. Other times she isn't really wanting to experience help because her experiences with treatment have not been good. In my study, I found 12 themes. Of those 12 themes, the very last one's the most important one. My participants expressed that they did not have a great experience with receiving treatment. What we can do for our individuals with eating disorders is provide appropriate treatment and provide appropriate care. We can't expect to eliminate eating disorders, but we can provide appropriate care. Thank you.
The title of her talk is Relational Health and Positive Psychology Characteristics as Predictors of First-Year Student-Athlete Adjustment.
So when you think about a first-year student-athlete, what comes to mind? First-year college students face a very unique set of challenges as they transition into the university setting. Academic challenges, institutional challenges, creating relationships with friends. First-year student-athletes are not very different. However, first-year student-athletes experience an additional layer of unique stressors when it comes to that collegiate transition. And for that reason, I decided to do my dissertation on relational health and protective factors as predictors of first-year student-athlete adjustment. In order to examine adjustment, I really needed to break it down because it's such a broad construct, there's so much involved in it, and I had to figure out a way to streamline. So what I did is I broke it up into 4 core areas: predicting academic adjustment, personal emotional adjustment, social adjustment, and institutional adjustment. What I wanted to know is does the relationship between coaches, teammates, and the institution impact adjustment for the first-year student-athlete? Beyond that, I wanted to know if personality characteristics like the presence of resilience, grit, and mattering impacted adjustment for first-year student-athletes. My results were kind of interesting. What I found is that when looking at academic adjustment, grit and mattering were the 2 variables that were statistically significant contributors. When I looked at personal emotional adjustment, resilience, grit, and mattering were all statistically significant contributors. When I looked at social adjustment, mattering and relational health with the university were 2 statistically significant contributors. And when I looked at institutional adjustment, the presence of grit, mattering, and relational health with the university were statistically significant contributors. So what does this mean? We have all these predictors. What do we do with that? What this tells me is that the presence of feelings of grit and the feelings of mattering for a student-athlete can impact adjustment on all 4 of the areas of adjustment that I evaluated. If these things are present, institutional adjustment increases, personal emotional adjustment increases, social adjustment increases, and academic adjustment. Beyond this, when I looked at those relationships, I found that relational health with the university, that connection that an individual feels to their institution, was a statistically significant predictor of institutional adjustment. They're a little more comfortable in their settings. And social adjustment, they're willing to meet other people and put themselves out there. With this information, I'd like to inform program development. The first-year student population, specifically with athletes, makes up a large portion of the student population. So with that, if we can create programs that inform the development and support of athletic adjustment, we can really make a difference for these students. Thank you.
The title of his talk is Faculty Sense of Efficacy for Culturally Responsive Literacy Instruction at Hispanic Serving Institutions.
When I was an undergraduate student, I remember walking into my first classroom. This was a big auditorium, a science class, and I was sitting with 150 other college students. And the first words of my professor said, for me, I teach all my students as if they were color gray. I don't care if my students are black, white, yellow, or brown. I teach all my students the same way, and despite their cultural differences. And when I heard those words, it was really shocking, because that is really, really bad. It's really important that you take into consideration your students' backgrounds and where they're coming from, because you need to understand, you need to know your students in order to teach them properly. So that was the main motivation for my study, my dissertation, because I analyzed the faculty sense of efficacy for cultural responsive literacy instruction at Hispanic serving institutions. And this is a publication from the Pew Research Center, and you can see here that on the undergraduate college completion, you can see that Asians and whites, they have really high completion rates, and then you can see the decline in minority groups with blacks and Hispanics. You can see that only 22% and 15% of Hispanics are actually finishing their career, their college degrees. So you can see that actually that decline is actually affecting the society, because you have less prepared individuals, and that's also affecting the universities, that's actually affecting funding. So you can see that's the biggest problem. So when I created the survey, we measure the writing, visualization, thinking, culture, and validation of the faculty sense of efficacy. And when the information that we got was that it is important that students feel welcome. It's important that you create a sense of environment that the students feel welcome, and that you measure this with the students. So when you bring the students, it's important that the students feel validated, and that students feel welcome, and the students feel supported. So it is important. You see nowadays in the news, you see everywhere that the government and the immigration laws are trying to build walls. So it is important to make sure that students, instead of creating walls, we need to create bridges. So it is important that the students feel welcome. Thank you.
The title of this talk is Ethanol in the Atmosphere.
Good evening, everyone. Unlike a few politicians and businessmen, I believe everyone in this room believes that global warming is real, and we humans are responsible for it. Global warming is caused when the layer of ozone closer to our Earth increases to form a blanket around our Earth, so that it gets trapped in the lower atmosphere and thereby increases our Earth's temperature. Various chemicals emitting from industries and factories help to form this bad ozone layer, which is also associated with several respiratory diseases like asthma and bronchitis. One of such chemicals is aldehyde. This aldehyde also has a potential of causing cancer. So where did this aldehyde come from? Well, when ethanol reacts with oxygen in our air, it forms aldehyde. Considering this fact, 64 countries in the world have already set a mandate for ethanol production. Unfortunately, the US is not one of them. The reason for this is we don't have a baseline of atmospheric ethanol to design such mandate for ethanol production. This is where my study comes to play. My study will provide a baseline of atmospheric ethanol in several states of the US, and also categorize the ethanol coming from plant source and that coming from human activities. This is very, very important in order to design a perfect mandate for ethanol products. Ethanol, as it is one of the renewable fuels, is being highly preferred as an alternative fuel for vehicular and industrial use these days. So to meet this increasing demand, the production of ethanol is also increasing exponentially in the US, as you can see in the graph. The problem is, the more we use ethanol as a fuel, the more we release it to our atmosphere, which then gets converted to either aldehyde or ozone. Again, neither we want global warming, nor we like cancers or respiratory diseases. So my study has potential to save our future generation from such kind of phantoms. So far in my research, all the samples have already been collected, and the initial analysis of a subset of data shows that we have very high atmospheric ethanol in several states of the US. Thank you very much.
This presentation is entitled "The Significance of Sustainability Reporting to the Realization of Sustainable Development."
I hope everyone believe, that is, my belief is everyone hopes to see the world as green as this someday. That is how I hope. But you will agree with me that in the business world, the focus of everyone is on making profit. We as members of the public, even when we want to make investment decision, what we consider is profit. Sometimes these companies, they make this profit at significant cost to the society. Some of you can still remember the impact of BP oil spill in 2010. In spite of that human and ecological catastrophe, BP made a substantial profit in year 2011. You can check it out. Thank God. Fortunately, following several protests, boycotts, some companies have started to report on the impact they are making in the society. In September 2015, the United Nations General Assembly launched 17 Sustainable Development Goals with the hope that, if these goals are realized by year 2030, the world will be a better place. I don't want to bother you about the details of those Sustainable Development Goals. I want to talk specifically about goal number 2. This goal called on companies to be aware of sustainable development. And in my study, I wanted to know whether there was a significant improvement in the reporting pattern of these companies after the launch of sustainable development. The purpose is, I believe, if there is a significant improvement, that shows that companies are committed. What did I find? What did I do? How could I grade this? Then to do that, I compared the sustainability performance of these companies of 10 global companies before the launch of sustainable development and after the launch of sustainable development. I found a significant improvement in the performance index reported. What is performance index? This is just an indicator that indicates the efforts of a company to make impact in a particular area. I found that there was a significant improvement in the number of index they reported. Each of the reports I examined contained a section that detailed the effort made by the company to identify, analyze, and respond to social and environmental challenges. This might mean that companies are committed. However, there is more to do. That somebody changes smoking habit does not mean that the health hazard of smoking is eliminated. Companies still need to do more. And we as members of society, we need to consider the sustainability practice of companies. Thank you.
The title of her talk is Antimicrobial Gene Expression Within Marine Sediments.
Good evening, everyone. All of us in this room are familiar with competition. Humans experience it, animals experience it, and even those organisms we can't see, microbes. So in this top figure here, you can see this is a petri dish with 2 different microbial growths. You also see this zone of clearing between them represented by this red circle. These microbes are about to fight it out because they are limited in that environment. How do they fight one another, you may ask? They can produce these special molecules called antibiotics. All of us should be familiar with antibiotics because we've probably taken them throughout our lifetime to rid our bodies of harmful infection. On behalf of the microbes, you're welcome. But with that being said, we don't know what exactly causes them to produce these antibiotics. It could be from a competition mechanism of lack of nutrients, lack of space, or they could be working together to fight some other organism in their environment. So that's where my dissertation comes into play. We're interested in looking at natural antibiotic production within an ecological perspective. Most terrestrial soils have been excavated already, and with water covering 70% of Earth, the natural place to look is our oceans. So that's what we're doing. In figure 2 here we have a drilling vessel that's from the International Ocean Discovery Program that can be used to access seawater as well as sediment thousands of meters below the surface. That's how we have access to our samples, and we've already collected samples from the Mariana Trench. To the right, you can see in figure 3, this is how we're going to look for antibiotics. Everyone has a genetic blueprint. All living organisms have it, including microorganisms. We can look specifically for those antibiotic-producing genes in order to determine if they can produce that molecule we're after. We can even take it a step further. If that gene is turned on, then it's producing that molecule, and it's producing RNA telling that cell, hey, let's make this molecule and compete with that other organism that's in our vicinity. So we can look for DNA and RNA within that system and try and determine if they're making that antibiotic. Now what we want to do with this information is construct this global map. What we can do now is we've already looked at deep sea sediments and seawater samples, and we've found that antibiotics are found in these systems. So that means it's not only in our soils outside, it's everywhere. We just have to look. So not only does this leave an opening for possible ecological discovery for what these organisms are doing within their natural environment, but also could lend a hand in medicinal discovery as well, with the rise of antimicrobial resistance. Thank you.
The title of her talk is General Self-Efficacy and Teacher Sense of Efficacy of Generation Z Teacher Candidates.
Good evening. In 2017, I read an article behind my desk as an administrator, and I read an article that said 6%, only 6% of administrators understand the needs of this new generation of teachers that are about to come out. And it said Generation Z, and I thought, Generation Z, just the term itself sounded odd. So when I looked up Generation Z, it was individuals born in 1995 or later. So it makes up this unique cohort. So we have these new teachers coming into the teaching profession, and so out of 1,700 that answered this administrator, still over 1,500 still don't understand. So I thought in my head, what is driving this? Why is this occurring? So there was a problem, which then led to my research question. What is the general efficacy and the teacher sense of efficacy of Generation Z teacher candidates? In order to get this problem solved or contribute, I administered surveys to Generation Z teacher candidates. And what it showed was exactly what the research does show, that pre-service teachers do possess a moderately high sense of efficacy. So it didn't give me the results I wanted. So therefore I took it a step further and I administered 2 focus group interviews with Generation Z, each one, 8 candidates. And it revealed some very unique themes, 3 of them. The first one that came out was caring. Gen Z is absolutely empathetic and they're caring. And when I say caring, they care about the educational field, they care about their own self-worth, and then they care about their students. But more than anything else, they care about the mental health. Not only the mental health of themselves, but the mental health of their students. Research shows today that mental health in this particular generation is on the rise, more so than any other cohort before them. they are cognizant and they want professional development on how to train students regarding mental health. The next one that came out was diversity. Next year, 74 million individuals will have been born in Gen Z generation. So they embrace diversity. 1 in 4 is diverse. They want it, they want to celebrate it, they want to engage in it, and they just want to be accepted. Not only themselves, but accepted for their students as well. The last major theme that came out was instruction. What are their beliefs about instruction? 2 things. General instruction has to have technology. Without it, there is no learning. My hope is that we provide PD. Thank you.
And the title of her talk is Assessing the Link Between Learning Modes and Social Media Use for Learning in First-Year Undergraduate Students.
Where can you go to learn how to play the guitar, fix your ice maker, and even sew on a button? YouTube. Did you know that YouTube is the most popular social media platform among all age groups? Social media use is pervasive in our society. Nearly 90% of college-age students report using social media. So why aren't we using it as a learning tool in higher education? The problem is there isn't much research on how to integrate social media into the classroom. What are the instructional considerations? How do students prefer to learn? To frame my research, I chose Kolb's experiential learning model, which is a cycle of 4 learning modes: concrete experience, reflective observation, abstract conceptualization, and active experimentation. Simply put, students learn by experiencing, observing, reflecting, thinking, and doing. I wanted to find out whether students' learning preferences influence their use of social media for academic purposes. So, I asked first-year learning students in a learning community to rank their learning preferences by selecting sentences that best describe them. Like, I learn best from rational theories, or I prefer to learn new experiences. I then asked them to select how often they used social media to support their academic learning. For example, I searched YouTube for a video to find information about a topic I'm studying regularly, sometimes, or never. Results showed that students' learning preferences aligned most with the concrete experience mode, which involves interacting with people and feelings associated with learning. Despite their preference of that mode, students more often use social media for independent tasks like searching for class-related information or storing useful apps on their smartphone. While these tasks are helpful for learning, leveraging the student's preference of the social aspect of social media should be further explored. With social media, students can collaborate with their peers using blogs and journals, or communicate with experts in their field using Twitter chats or Facebook groups. Integrating social media into the curriculum prepares students to better live and work in an ever-changing world. Thank you.
The title of his talk is Hydrologic Controls on CO2 Flux in 4 Texas Coastal Estuaries.
Good evening. So I think my research topic is more related to climate change because we all know carbon dioxide, the CO2, is a greenhouse gas which is responsible for the climate change, right? The coastal system is usually studied as a CO2 source to the atmosphere because of a lot of carbon from terrestrial or human activities would be flushed by the water into the river and finally get into the coastal zone. Unfortunately, Texas coast has one of the largest CO2 emissions among all the North American coasts. Agreed or not, we are experiencing more intense climate change during the last few years. Our coastal system is facing more severe flooding situation due to hurricanes, thunderstorms, or winter cold. So my research is to explore how these flooding situations could change the coastal CO2 level. So during the last 4 years, from 2014 to 2018, our team went out to the base, sampling from northern Colorado coast to the southern Corpus Christi coast, and we took water samples at least every season. Our data shows a very clear trend of increasing CO2 level in response to those floodings. For example, during 2015 summer flooding, we observed the immediate decrease of CO2 level. Also, during Hurricane Harvey, we observed another strong CO2 signal, especially for the Guadeloupe coast. So the flood could strongly increase the CO2 level in the coastal area. Just think about the coastal system like a human body. When you eat a lot, you have to digest it and exhale a lot of waste to balance your body system, right? So the ecosystem is the same. Our conclusion is that the increased CO2 level in coastal area could be expected if the flooding situation gets more often in future. This increased CO2 level will in turn to feedback the climate change to make it more severe in future. Our biggest suggestion is to carefully design the coastal water management in future, which should include most of the severe situation. And that's my talk today. Thank you.
The title of his talk is Harmful Microbe Farts. Yeah, it's like that. Okay, and how they can be stopped.
Hello everyone, I'm here to talk to you about harmful microbe farts. There are many types of farts from microbes, but the one we are most interested in is methane. Methane is a very potent greenhouse gas. It's the second most important greenhouse gas in the atmosphere. But the good news is that it has a very short lifetime in the atmosphere, which makes it a good option for us to mitigate. So if we can stop methane emissions to the atmosphere, this will have more of an immediate effect on global warming. The producers of this methane gas are these microbes over here that we like to call methanogens. We'll call them methane makers. When you think of methane production, you might think of either like cows or maybe termite guts, but what is actually going on is it's that these methane makers within these cows' guts or within the termites' guts. So these are like the biggest cause of methane production. They take CO2 and they will form it into methane. On the other end of this equation, we have our methane eaters, methanotrophs. So they will take this methane in the presence of oxygen and form it into CO2. And so it forms this cycle. And that's why it's so hard to try to understand what's going on with methane production. When we go out into the field and measure, like let's say out into Oso Bay, and we want to measure methane production and we see that the levels are very high, does that mean that there's very little methane consumption, or is there just so much methane production, and what's affecting these relationships? So to start breaking down this cryptic cycling of methane, we want to look at our methanotrophs first. We want to look at them and how they work in culture under various oxygen conditions, especially low oxygen conditions, because we want to be able to work with these methanotrophs, methane eaters, and our methane makers together. And if that's going to happen, they have to kind of work in this middle ground of environment with low oxygen for the methane makers and enough oxygen for our methane eaters. We then want to measure our cell activity of the methane eaters, so that way we can work on further quantifying what's actually going on with this process. And then the third step is that we want to introduce the methane eaters and the methane makers in order so we can try to study this relationship further and break down this process of what affects it, So that way we can make better predictions of what tomorrow might look like with the changing climate system, as well as effects that we may have on this environment.